Thank you for introduction, and thanks to the INC Academy for this wonderful opportunity to present today. So my story is about a novel and functional role for architectural proteins in 3D chromatin organization in Drosophila. And before going directly into the results, I want to start from a very brief introduction to key ideas of the chromatin architecture. So we have a long DNA stored inside the tiny nucleus. So some DNA fragments will be in close spatial proximity and will interact with each other. We believe that there is a specific chromatin architecture, so some fragments interact more frequently than others. For example, distal enhancers are brought closer to their target genes to achieve effective gene expression. So should be a connection between 3D genome organization and cell function. Hi-C is a widely used experiment that helps us to study the interactions happening between different DNA fragments. During the experiment, the population of cells is exposed to formaldehyde, and fragments which are in close spatial proximity will be cross-linked. So the count data then is written in the form of the matrix, where each matrix entry is being an interaction between row locus and column locus. In the Hi-C contact map, we observe high interaction intensity close to the diagonal, and interaction in this intensity decreases when we move further from the diagonal. We clearly can see the intensive squares along the diagonal, and these structures are named as topologically associated domains, or shortly TADS. And TADS is literally a cloud of chromatin stuck together with intensive interactions happening inside. TADS are widely conserved across species and as well as during different developmental stages, suggesting that they have a functional role. In Drosophila, several architectural proteins occupy TAD borders, suggesting that they play important role in TAD organization. The list of such proteins contains, first of all, CTCF and cohesin that are widely known as architectural components in mammalian systems. Then there is a BIF32 that demonstrated strong enrichment at TAD borders in cell lines derived from the embryos. Two more proteins are CP190 and chromator. They cannot bind independently to DNA. They are recruited mainly by BIF32, and it was shown that up to 90% of TAD borders in Drosophila S2 cell lines were occupied with BIF32 together with CP190 or chromator. Another interesting protein in Drosophila called DREF, and it displays the same binding motif as BIF32. So it's one possibility that DREF can occupy TAD borders when BIF32 is depleted. The aim of the current research is to measure the changes in 3D chromatin organization together with change in transcription to dissect the formation mechanism at TAD border and evaluate the functional role of TADs. To do so, we depleted BIF32 in BG3 cells derived from the larval central nervous system and also performed double knockdowns of first CP190 and chromator and second BIF32 and DREF, using RNA to disentangle the interactions between different architectural proteins at TAD borders and their overplay with each other. Western blots demonstrated that the knockdowns lead to specific and strong reduction in the aimed protein levels. And in terms of the numbers, protein levels were reduced by around 70%. This is efficiency achieved. This efficiency achieved is similar to the ones reported in. Another important thing here is that knockdowns showing no cell cycle arrest. Previous studies demonstrated that TAD organization undergo dramatic structural changes every cell cycle. So the FACS analysis ensures us that the changes that we observe in TAD structure are related to protein depletion directly and not caused by cell cycle arrests. Before going to TADs, we want to look at changes happening at higher genome organization level. And on the top panel, you see the part of Hi-C generated matrices reflecting the chromosome 2L. So BIF32 knockdown results in loss of long-range interactions and showed an increase in short-range interactions. Likewise, CP190 chromator double knockdown as well as BIF32 double knockdown shows the same. But the loss of long-range interactions was less pronounced compared to single BIF32 knockdown. Previous analysis of Hi-C data has shown that the genome can be divided in two compartments called A and B compartments. The DNA fragments belonging to the same compartments are specially colocalized, and these compartments are associated with open chromatin for A compartment and closed chromatin for B compartment. Our research confirmed that there are negligible changes in the proportion of the genome that is in A or B compartment in all knockdowns. Switching happened only in around 5% of the cases. So if you look on the eigenvalue profiles, like these profiles are used to identify affiliation to A or B compartment. And you can see like between all the mutants and wild type, they are approximately the same. Next, we looked closer at the next organization level, its TADS, and we identified between 2,000 and 2,600 TADS at DPN2 resolution, so it's for average bean size of 5,000 base pairs. Depending on the strength parameter, we defined both strong and weak borders, and then we looked at differences between wild type and knockdown separately. The most interesting groups are maintained and lost borders, and we will focus the further analysis on these two groups. The mechanism of obtaining new borders as a result of protein depletion seemed intriguing, and after careful analysis, we found that the majority of these new borders just a move from like happen more than 2 kb away from the wild type TAD borders comparing with the knockdowns. Only a small proportion of new TAD borders are actual new borders, so results in splitting the original TAD into separate TADs. The following examples nicely represent the described scenarios. So on the left panel, we see that some of the borders are allocated at exactly the same places, both in wild type and in the mutant. So as before the protein depletion. Some borders, like in the middle panel, are lost and results in aggregation of neighboring TADs into one big TAD. And on the right-hand side, it's a new border that mainly just a large shift of existing TAD border in wild type, but sometimes maybe a real new border that splits the existing TAD into two subTADs. Compared to wild type, out of all strong borders, 188 borders were maintained and 149 were lost in two knockdowns. The rest of the strong borders in wild type cells either not displaying the same trend in both knockdowns, moving only within 2 kb and defined as fuzzy, or only moderately weakening. So we wanted to be sure that the borders that we focus on are direct targets, meaning that before the depletion, the borders were occupied by the proteins of interest, so BIF32, CP190, or chromator, and then these borders maintained or lost due to direct effect of the depletion. Majority of maintained borders are direct targets of the three proteins, but only half of the lost borders are direct targets. To further confirm that direct maintained and direct lost borders are indeed controlled by the three architectural proteins, we analyze the ChIP-seq data. So we analyze the ChIP peaks near the direct TAD borders. And we found that the majority of maintained TAD borders retain BIF32 or CP190 upon knockdown, while most of the lost borders lose binding of these architectural proteins after knockdown. To determine the chromatin and epigenetic mechanisms presented, maintained, and lost borders, we analyze the presence of key factors such as architectural proteins, transcription, replication, and accessibility-related complexes. So here you see the heat maps of the ChIP occupancy profiles constructed within 5 kb window around the direct maintained on the top and lost TAD borders on the bottom. So let's have a look on some specific examples. So CTCF was partially presented at the maintained borders, approximately at half of the borders. But there was a strong enrichment of cohesin at majority of maintained TAD borders. So we see the cohesin subunits RAD21, NIPT-B, and SMC1. Also, we see significantly lower histone levels like H1, H3, H4 at maintained borders, indicating the presence of highly accessible DNA. The maintained borders were also enriched with POL2, mediator complex, and ORC2, plus strong divergent transcription at the maintained borders. In contrast, at lost TAD border, there is a less DNA accessibility and transcription, indicating that these borders are in repressed chromatin state. So also we investigated the presence of histone modifications, and we found that the majority of so the maintained borders are found to be enriched with active promoters around marks like H3K4 free methylation, enhancer mark that is H3K4 monomethylation, and together with H3K27 acetylation. The lost borders were strongly enriched with mainly signatures for heterochromatin and polycomb. However, we did not observe significant enrichment for any polycomb subcomplexes like PC or D-ring in the two knockdowns. Nevertheless, we did find enrichment of SU-VAR39 and HP2, which explains the enrichment of heterochromatin at low stat borders in the two knockdowns. So in simple words, we observe that maintained borders are chromatic, while lost borders are heterochromatic. Surprising thing here is that previous research reported that TAD borders are mostly composed of L-chromatin in flies. Using a chromatin state map in BG3 cells, we further investigated the chromatin states associated with maintained and lost TAD borders, and our results confirm that indeed maintained and lost borders are enriched in enhancer and active TSS chromatin states and are depleted in heterochromatin. In addition, lost borders also display partial enrichment in polycomb state. The apparent differences in results at lost borders can be explained by the fact that chip profiles were constructed over a 5 kb region, while the chromatin state analysis was performed at the base pair resolution. So if we consider the same 5 kb region, the results will be consistent with the chip profiles. This means that while the majority of borders are enriched in enhancer or active TSS, maintained borders are located in L-chromatic regions and lost borders in L-chromatic islands in heterochromatin. So we also look at the gene expression analysis to see is there a connection between TAD organization and gene expression. And first of all, we did not detect any differentially expressed genes that are allocated exactly on the border in either wild type or mutant or both. So all the differentially expressed genes belong to inner TAD areas. And plus, the differentially expressed genes did not demonstrate any preferences towards specific localization near or away from TAD border, meaning that genes are spread approximately uniformly within TADs. We group genes depending on 4 different TAD reorganization patterns, like you see in blue. And the majority of differentially expressed genes are upregulated and belong to the two largest groups, that is consort1 border and knockdown specific borders, that can be defined as a massive TAD rearrangement. So for example, on the right-hand side, you see the panel that show like how knockdown specific border change look like. So we see many aggregations and some border shifts happening simultaneously. We also perform a permutation test that showed that we see the statistically significance between differentially expressed genes and reorganization of TADs only in case of single BIF32 knockdown and BIF32 and DREV double knockdown. And these associations were significant only in case of these largest two groups, so meaning that in case of massive TAD rearrangements. So we also looked at the next chromatin organization, that is a Chromatin Loops, and out of 770 loops that were detected in wild type in each knockdown, approximately 200 are maintained and 300 maintain only one anchor in the same position. So here you also see the aggregated peak profiles over the maintained and lost chromatin loops. And it confirmed that the strong level of interaction is maintained in the two knockdowns at only maintained chromatin loops, while it's not the same in case of the lost loops. The maintained loops display higher level of chromator errors compared to the lost loops, suggesting that the depletion of chromator or blocking of its recruitment, that is a BIF32, is not sufficient to affect the maintained loops. The majority of chromatin loops in our dataset are located near a MED1 chip peak, indicating that mediator complex would be more important for chromatin formation in Drosophila. So also a small note here on gene expression, and there is a statistically significant difference in the number of differentially expressed genes at maintained and lost loops. So for example, you on the right-hand side, you see very nice examples of the chromatin loops, like on the top, where we can see the chromatin loop that is maintained in 2 knockdowns while the target genes is differentially expressed. Or conversely, on the bottom panel, we see the lost chromatin loop that can lead to no changes in gene expression of the target gene. So these results support a model where the presence or absence of a chromatin loop does not necessarily lead to the regulation of the target gene. So we have more associated analysis and figures that expand the story and look at some results deeper. This manuscript is currently under revision and is not published yet, but if you're interested to look at the research more carefully and find some specific aspect of the result that you're interested in, please have a look at our bioRxiv preprint. As we approach to the end, I want to summarize the most important takeaway messages here. And so first of all, protein knockdowns result in reduction of long-range interaction and increase in short-range interactions. There are negligible changes in AB compartments in all 3 knockdowns. That borders are maintained by architectural proteins, divergent transcription, and associated factors. At lost borders, there is a less DNA accessibility and transcription, indicating a repressed chromatin state. Maintained borders are located at L-chromatin and lost borders in L-chromatin islands in heterochromatin. Many large reorganization of TADs correspond to significant changes in gene expression, and this is not the case in the situation with the chromatin loops. Majority of chromatin loops are controlled by mediator complexes, chromator, and CP190. So here we are at the end of my talk, and I want to thank everybody who participated in this project and supported us. Special thanks to Dr. Kitty Chatot, who generated high-C data that we analyzed and presented today. Thanks for support to all members of Zabet Lab, especially to Jared Wolf, who produced summary analysis, and Dr. Radu Zabet, who supervised the project. Also thanks to School of Life Sciences and Department of Mathematical Sciences in the University of Essex for interesting discussion and ideas to develop and improve the results. Also thanks to Drs. Hugh Cotterill and Jill Crever from St. George University, London, who helped us with additional lab experiments during the pandemic times. And last but not least, I want to thank for your attention and thanks to INC Academy once again for this great opportunity to present today. And we are happy to answer any questions about the current research and any kind of questions. Thank you very much for your attention.
We have one question from Elizabeth Ing Simons on the Q&A box, so I can read it. Maybe it's visualization issue, but to me, the high-C matrices and log2 control versus knockdown you show don't support the loss or gain of TAD boundaries at the regions shown. Apart from the TAD boundary calls, have you checked if there is significant changes in insulation scores or directionality index at this region? Since it is a K results, it would be good to validate it with other approaches. It's a long comment and question.
Yeah, I see. Give me one sec to properly answer this question. So when we looked at the TAD borders, yes, we called the TAD, first of all, we called the TAD borders by high-C Explorer using 2 different threshold measures. So we ensure that we call the TAD borders that have a higher separation score called as a strong TAD borders, and also we called as weak TAD borders to see what actually the difference between them and how the changes can be affected by different thresholds. But as well, we also did, yes, we did the, we plot the distribution of directionality index to support what actually changes we observe in TAD boundaries. So yes, visualization is just a very nice way to represent, like to show the result and feel the difference, but also we produced some plots to actually have a look on the distribution. So we ensure that our changes in TADs are significant.
Hi. Okay, thanks for your talk. Very interesting. I was just confused about one point. Maybe you can clarify. So if I understood correctly, so when you knock down these proteins like chromate or CP190B, right, which are enriched, their binding is enriched at housekeeping gene promoters, right? So in euchromatin, but you're losing the heterochromatic boundaries. So what was the model again to explain why it's not the euchromatic boundaries that are being affected?
Yeah. So when we wanted to look on the occupancy, we constructed the chip peak around 5 kb window, because if we have a TAT border, it actually can fuzzily move. So we expect that it will be not like exact position, it will be some window. So when we looked at 5 kb window, we construct a clustering analysis. So we just look on the general signal inside the window. So that's why we observe the heterochromatin marks at lost borders. But if we look at base pair resolution, like at each exact position, so we will observe the result that is consistent with previous research, that is mainly euchromatic regions. So meaning that if we look on the bigger perspective, on the bigger window, we will observe heterochromatin. While looking on the base pair, we lose euchromatin regions. So that's why we expect that we lose the lost borders allocated in euchromatic regions, that is like euchromatic islands within the big heterochromatic regions.
Hello. I'm not a great specialist in this method, but in general, I have two questions. TATs are known as a conservative units, yeah, conservative through the animals and so on. So, do you doubt it, or do you find the thing that these changes, now if they may be also natural, are can be referred to more conservative genes or to housekeeping genes, not housekeeping genes? Do you consider this thing? That's the first question.
It's interesting question, and I will be completely honest, because we looked on mainly on the Drosophila, we just compare like different cell lines. And we didn't look exactly on the, like we didn't look deeply on the genes that we like observed there. So I personally will not say something like very strict. So I actually not sure, and it's like a nice direction to have a look on. So
Now, if my question is not silly, I'm not sure, but okay. Now the second question. What are these euchromatin islands in heterochromatin? If we imagine nucleus morphology, what is it? It is interchromatin spaces, these are spliceosome regions, or what these are, what they are referred to?
it would be very nice to like visualize it and have a look like how actually in space it will look like. The only thing that we can currently say based on the results of the research, we just observe some like some marks that identify heterochromatic and euchromatic regions. And without like proper visualization, I don't want to have a strict answer for this. So
okay, thank you very much.
It's a nice ways to like to expand further and visualize it and see how it looks in 3D.
If you know, give me a note. Thank you very much. Okay.